So hi everyone. My name is S Siu, and I'm a research staff member at the IBM TJ Watson Research Labs in Yorktown Heights, New York. And so today I want to give a presentation on the work that our group has done to create a quantitative point-of-care digital urinalysis device for nitrite detection. So why do we need a quantitative point-of-care digital urinalysis device? There are over 2 billion urine tests performed globally every year. Most urine tests are performed at home or in the clinic using dipsticks, which are highly subjective and qualitative. Currently, quantitative measurements for urinary analytes such as nitrite, protein, and creatinine, and others would actually need to be done in a clinical lab using large benchtop instruments. Since these laboratory tests are done infrequently, this data that they collect only provides a snapshot of a person's health and time. So if a portable at-home quantitative urine testing device is available, more frequent testing can be done, and the envelope of quality care can be extended outside of the hospital, resulting in earlier disease detection and/or medical intervention. So in this presentation, we discussed the first prototype of our quantitative urinalysis device for measuring nitrite. And nitrite is an important biomarker to predict urinary tract infections, since approximately 15 million women in the US suffer from recurrent UTIs, and it costs about $4 billion in emergency admissions and just in dealing with UTI-related issues. A healthy individual should not have nitrite in their urine, so it's a good biomarker for UTI detection. If an individual actually has a UTI, the bacteria converts a nitrate that's in our urine into nitrite, and so you would pick that up. Other biomarkers such as leukocytes and pH can also improve the sensitivity and specificity of a test to predict the onset of UTI. So we compared the performance of our digital urinalysis device with that of two commercial dipsticks. So we used the ChemStrip from Roche Diagnostics and the multi-stick dipstick from Siemens Healthcare. It is known that dipsticks are highly subjective, qualitative, and its accuracy is highly dependent on the volume of urine that's applied to the pads, the lighting in which the dipstick is read, and also the reaction time. So when does the user actually read the dipstick? The image here on the left are examples of the Roche and the Siemens dipsticks dipped in either 0 or 200 micromolar nitrite solutions. And you can see that the dipstick pad turns pink in the presence of nitrite, so in both cases. And this is due to the grease reaction and the formation of a pink azo dye in the presence of nitrite. We will discuss the chemistry in a bit more detail in a few slides. So the figure here on the right shows an array of nitrite pads stitched together from cropped images of independent ChemStrip and multi-stick dipsticks. The top 5 rows uses nitrite spiked from 0 to 200 micromolar in a salt buffer at varying pHs from 4 to 8. And then the next row is nitrite spiked in artificial urine. And then we also use healthy normal urine from human subjects. So there's 2 examples here. The dipsticks in this figure were actually read by a reflectance analyzer. So the Roche has a URIS analyzer and the Siemens has a Clinitech Status Plus. And what the analyzer reports out is either a negative or a positive. So their readout is just negative or positive. And if the sample contains enough nitrite that the analyzer can read, it will give a positive readout. And so you can see here, as denoted by the red boxes, the commercial dipsticks will reliably detect nitrite at 25 micromolar or higher. So here's an image of our transmission quantitative digital urinalysis, or in our case, we say colorimeter. So we do use colorimeter and digital urinalysis device interchangeably through this presentation. This colorimeter is a handheld single channel device that's housed in a 3D printed casing and pairs with a mobile application to measure the amount of nitrite that is present in the solution. Samples are measured using a 10 millimeter path length polymethyl methacrylate, so PMMA cuvettes, which they have an optimum transmission in the visible spectral range from 340 to 800 nanometers. A user can add their urine samples to a cuvette containing nitrite detection reagents, mix, and then place the cuvette into the device for reading. This device has a built-in interference and color correction algorithm to collect for the color of the urine. And compared to the commercial dipsticks, this device has been found to be more sensitive and has a broader detection range for nitrite. And the overall time to run the assay can be less than 5 minutes, and it is portable and affordable, making it suitable for at-home monitoring use. So here are two schematics showing the top and bottom views of the device and the placements of its internal components. So here's the sample slot where we put the cuvette in. So in the sample slot, there is a pair of multi-wavelength LEDs. So it's an RGBW LED. And then directly opposite of it is a photodetector for measuring transmission measurements. And then at 90 degrees to it is another photodetector that we have for measuring turbidity measurements, looking at the scattered light from the sample. So we use a surface mount RGBW NeoPixel LED from Adafruit as the light source, and this was selected for its small form factor and digital control. An Arduino controller board with low energy Bluetooth capabilities, such as the Adafruit Feather MO, was used to control the operations of the LED and the photodetectors. It was also used to communicate with an Android smartphone application for data collection, analysis, and storage. The Android application can display the concentration of nitrite for a single event, but also for a set of readings over time. So here is a cross-sectional picture of the sample slot. So you put the cuvette in here, and then there's the LED and then the photodetector. The LED is at a fixed height, and so we had to perform an experiment to determine the minimum reaction volume needed to reliably measure a low and a high concentration of nitrite in solution. And so we just did different volumes, and we found that the minimum reaction volume in the cuvette needs to be 0.8 milliliters. So moving on to the nitrite detection chemistry, our team looked into the literature for various methods to detect nitrite in solution. We found that the grease reaction is a common method for detecting nitrite. However, there are many permutations of the grease reaction that's currently used in commercial testing. So for example, reaction 1 here is the grease reaction that has been established as the European standard for determining nitrite concentrations in drinking water. So the grease reaction consists of an aniline derivative and a coupling reagent in an acidic solution. The most common arrangement uses sulfoamide and N1-naphthoethylene diamine dichloride, dihydrochloride, or NED for short. And when a sample containing nitrite is added, so here's nitrite, when it's added, the nitrite ions actually react with the sulfoamide in the grease diazotization reaction to form a diazonium salt. So here is the diazonium compound. And this salt then reacts with NED to form a reddish pink azo dye that can be quantified using a spectrophotometer to determine the nitrite concentration. So some common aniline derivatives include P-arsenic acid, nitroaniline, and P-aminoacetophenone, and other coupling reagents such as 3-hydroxyl 1234-tetrahydrobenzene quinoline, or THBQ for short, can be used to change the sensitivity and solubility of the azo dye formation in solution. So reaction 1 is the European standard for looking at nitrite concentrations in drinking water. And reaction 2 is the nitrite detection that's used on the ChemStrip by Roche Diagnostics. As you can see, both reaction 1 and 2, they use the sulfoamide as the aniline derivative. But in reaction 2, they use a different coupling reagent, THBQ. So reaction 3 that we looked at is the nitrite detection for multi-stick, dipstick by Siemens Healthcare. And so they also use THBQ, but they used P-arsenic acid for the aniline derivative. And so we also looked at reaction 4, which is a permutation of 1 and 3. And so instead of, so this one, for this grease reaction, we used P-arsenic acid and an NED. So while the grease reaction is well known for detecting nitrite in solution, we are not aware of studies that characterize and optimize the reaction for use in transmission measurements of urinary nitrite in a clinically relevant range. So we tested all four reactions described just now in a salt buffer containing varying concentrations of nitrite, 0 to 200 at pH 4.3 and pH 7.9. And we notice, you can see here, these are the cuvettes from 0 to 200. Reaction 2 and 3, actually, that uses THBQ, had a brownish hue, even at the lowest concentrations, and was not as sensitive as using NED as the coupling reagent. So part C here, the photos taken are of the bulk reaction of a solution, so reaction 1 solution and reaction 4 solutions at day 1 and also day 14, where we stored the solution either at room temperature or at 4 Celsius. We noticed that reaction 1 solution turned pink over the 2-week period, perhaps due to some impurities in the sulfoamine powder. So since the solution for reaction 4 containing p-arsenic acid and NED remained colorless over time, it was chosen and used as the primary nitrite detection scheme for subsequent experiments. We also looked at the reaction kinetics of reaction 4 in 4 samples containing different amounts of nitrite. So while the reaction was very rapid, you can see the sharp drop here. In theory, we can choose the measurement time anytime really after like 60 seconds. We allowed all reactions to go to 10 minutes to ensure that the reaction reached full steady state. And it is important to note that in this graph, we are using the transmitted intensity for the green LED instead of just the red, blue, green, and white. And the reason for that is we selected the green LED based on this experiment where we looked at the RGBW transmission curves. Here are the 4 plots of the transmitted intensity. Here's the white, the red, the green, and the blue. And we basically measured the transmitted intensity of the LEDs in samples containing varying concentrations of nitrite, ranging from pH 5 to 8. And we found that the green LED was the most sensitive to any variations in nitrite concentration. And this makes sense since the azo dye product formed from the grease reaction causes the solution to turn pink. And so the maximum absorption peaks would occur in the green wavelengths. So next, we wanted to validate our detection reagents from reaction 4. So we validated them against a nitrite detection kit from Sigma using the SpectraMax benchtop spectrophotometer. So again, samples were prepared containing nitrite ranging from 0.78 micromolar to 100 micromolar in a salt buffer at pH 7. And so the Sigma kit had a suitable nitrite detection range from 6.25, you can see by the red circles, and it's also denoted by the yellow box here. And the detection reagents for reaction 4 were well correlated with the kit when we used the spectrophotometer, as you can see by the black squares. So next, we made measurements on the colorimeter and also the spectrophotometer using samples with known nitrite concentrations and determined the concentration of the nitrite using the calibration curve. So both the colorimeter and the spectrophotometer detected nitrite between 3.125 to 100 micromolar and were well correlated. So this suggests that the colorimeter could be used for quantitative measurements, and coupled with its lightweight and portable form factor, the colorimeter could be translated for at-home use. So next, we looked at the performance of the reaction 4, and we carried out experiments to detect nitrite spiked in several solutions, a salt buffer, artificial urine, and also human urine matrices. We prepared serial dilutions of nitrite samples from 0 to 200 in a 5 millimolar hydronium salt buffer from pH 4 to 8. So you can see on the top graph. The samples were added into the PMMA cuvettes containing nitrite detection reagents and reacted for 10 minutes. And so you can see here are the cuvettes going from low to high nitrite concentration and the color of the reactions after 10 minutes. We found that the detection limit of the reaction 4 in a salt buffer solution was 0.8 micromolar, which is 31 times more sensitive than the commercial dipsticks. We also prepared similar spike samples in artificial urine, which was a ready-made solution that we purchased from Pickering Labs. The artificial urine solution was chosen since it closely resembled human urine. And we generated a new calibration curve using samples of known concentrations and used a calibration curve to determine the concentration of unknown samples. The bottom graph here is a plot that looks at the measured value versus the known concentration of the nitrite spiked in these samples. The 45-degree diagonal line is a reference line that shows perfect equality between measured and known concentrations. So as you can see here, most of the values lie very closely on this 45-degree line, and the readings from the colorimeters can be used to determine nitrite concentrations in unknown samples with good accuracy. In artificial urine, we found that the limit of detection was 1.6 micromolar, and so that's about 16 times more sensitive than the commercial dipstick. And you can see here the yellow box shows the detection range of the dipsticks. So at 25 is where they can start detecting. So the next feature that we had incorporated into the device is a color correction algorithm. So urine can vary from a light pale straw color for a healthy individual to a dark cloudy appearance in someone who is dehydrated or sick. Urine can also appear pinkish, red, or even blue depending on the food or medication taken. The color of the urine will impact the transmission measurement and in turn affect the accuracy of the nitrite detection. So it was necessary to develop an algorithm to remove the color effects from the nitrite measurements. And so to demonstrate the color effects, what we did was we added a water-soluble yellowish brown dye into the phosphate buffer and prepared solutions at a half, a quarter, an eighth, and a 16th dilution. And the nitrite was spiked into the diluted dye solution starting at 200 micromolar and serially diluted to 0. A sample without any nitrite was also added as the control. And so the top graph here is a plot that shows the measured versus known nitrite concentration before and after color correction. So the uncorrected are the open shapes, and then the corrected ones are the other solid shapes. So we found that the uncorrected samples can have data deviating from the 45-degree line, and the color correction reduced the error in nitrite detection by as much as 15 micromolar, which is significant, especially for lower levels of nitrite that's in the sample. So next we looked at the performance of the colorimeter and the two dipstick analyzers in nitrite spiked in 4 freshly collected human urine samples. So here is sample 1 to 4. And in normal, you can see these are the samples, and there's some slight, yellowish, depending on the actual human sample given. So we applied the color correction algorithm on these human urine samples, and it showed an improved correlation between the measured and the known concentrations of nitrite. 3 of the 4 human samples have nitrite values that were slightly under measured. You can see there's a slight deviation from the 45-degree line. And this actually suggests a potential interference from the sample matrix. It was subsequently found that the presence of urobilinogen in the sample can actually reduce the measured transmitted intensity. And so this is something that we would like to investigate further. We found that the detection limit in human urine was 6.4 micromolar, which is 4 times more sensitive than the commercial dipstick. Next, in order for the assay to be practically used, the nitrite detection reagent should be stable at room temperature or a refrigerated environment for a prolonged period of time. So initial stability studies were conducted by preparing and storing the nitrite detection reagents at room temperature, 4 degrees and -20, and tested over a period of 25 days. The nitrite readings remained stable for at least 25 days, regardless of storage temperature. So further work will investigate the ability to lyophilize the nitrite detection reagents and to determine the stability of the dried formulation. And then we also have a mobile app, and these are screenshots from the mobile app. And so the nitrite values are displayed on the mobile app after each measurement. The data are stored locally on the mobile phone, and the results can be displayed as a function of time. So here you can see the graph of trending nitrite, and they're increasing over time. And this tracking is important because if you can track how the nitrite changes from an individual's baseline over time, it can suggest that there may be a potential infection and ask the user to consult a physician. The application is programmed to highlight when nitrite levels are normal, right? And then display a warning message if the nitrite levels are getting too high. So in the next iteration of the colorimeter, the goal is to migrate all this data analytics and the storage of the data onto a HIPAA-compliant cloud-based server, which would allow for the measurement data to be easily accessible by clinicians or other members of the patient's healthcare team. So in summary, there are several methods for detecting nitrite in urine, such as the dipstick, the reflectance photometer, and even the large benchtop spectrophotometer. The colorimeter that we presented here today is in the third column, and it's a transmission-based method, and it can measure nitrite in a quantitative manner instead of giving a positive or negative readout. We found that the device is 4 times more sensitive than the commercial dipstick in human urine, which can lead to earlier detections of UTI and hopefully lead to earlier treatment. The device is built using off-the-shelf components and is roughly about $80 and is small and portable for at-home use. So with that, I would like to acknowledge and thank the team here that's listed here, which comprises of other IBM staff scientists, physician grad students, and summer interns for their hard work on this project. I would also like to thank the organizers of this webinar for the opportunity to present my work here today. I'm happy to answer any questions, and my contact email is listed on this slide. And if there's any interest in learning more about this technology, we do have a publication in ACS Omega that you can visit here. So thank you all for listening and for your attention.